This is a very general introduction to EEG, focusing on why we have such brain oscillations in the first place. So you measure EEG by putting electrodes all over the head. There are standard arrangements for that. You then get a trace for each individual electrode. Then you use software to try to localize where a particular oscillation occurs in the brain. For example, if you had such something happening here in the right prefrontal cortex, it would probably show up on these nearest electrodes and perhaps a little bit less elsewhere. EEG isn't very good at localization, but it has extremely good time resolution. So separating events in time, EEG is very good at, but localizing them to a particular part of the brain, that is more difficult. One important thing is that you can only get these traces here and see something more than a totally random squiggle if you have large numbers of neurons in the range of millions to many millions becoming active at about the same time, so in a synchronized manner. If all the neurons were working independently, you would only have something random if you recorded many millions of neurons simultaneously, which is what you do with EEG. So EEG is quite different from unit recording, where you put an electrode inside the brain and where you have the possibility of recording, of identifying the activity of individual neurons. EEG can only go for the simultaneous activity, the coordinated synchronized activity of very large numbers of neurons. And so then one question is, what are the benefits of such synchronized activity? Why does it exist in the first place? I know of two functions. There may well be more because this is not my field of research, so my knowledge is very limited. So I can only talk about those two, namely that oscillations offer you a different neural code that can be faster and can group and separate pieces of information, and also switching of information streams at a larger scale. So I'll be looking at what is phase coding, how can you get oscillating output from a steady input, how do theta and gamma oscillations interact, or how can they interact, the significance of phase and coherence, and the speed of NMDA channels. So one thing about codes is that in the old textbooks that and the things I was told 30 years ago in my first year's lectures was a frequency code, that information is encoded by how fast a neuron fires, the rate at which it fires. And that the dynamic range of a neuron is from about 1 Hertz, one action potential per second, up to a maximum of about 1,000, so that a neuron can code across 3 orders of magnitude. Now, for statistical reasons, if you want to estimate a rate, you actually need a reasonable number of events. And Gallistel, in one of his books, went through the mathematics and says you need 5 to 10 events in order to estimate a rate. So if you had a neuron actually firing at a rate of 1 Hertz, you would need 5 to 10 seconds to estimate that firing rate. And if that neuron is involved in determining where you put your foot so that you don't fall over, 10 seconds is probably a little bit too long. So you really need something a lot faster. And one code that gives you the option of being a lot faster is the phase code. This here is an image of place cells for which O'Keefe got his part of the 2014 Nobel Prize. So the idea is you put a rat, for example, into a rectangular environment and you record, so this is now unit recording, from the hippocampus, and you will find cells that are active in particular places. So if you think of each of those rectangles as a separate map of the same environment, so all of them show the same environment, but they show the activity of separate cells. So you identify a cell and you ask, where does this cell fire? And you map that onto the environment. And here you find one that is most mostly active in this upper left corner and a little bit here. Here is a cell that is active here on this lower edge. Here's another one also on the lower edge, but a bit further to the right. Here again, we've got that corner. Here again, the lower edge. Here we've got the right wall, and so on. So if you then have access to enough of these cells, then you can tell from the activity of these cells where you are or where a rat is. So already, I think in 1993, McNaughton did that recording here from multiple place cells for 15 minutes. Then for the second 15 minutes, again recording, but using the mapping from the first 15 minutes to work out where the rat should be according to only the about 100 place cells they recorded. And even with that small subset out of the total number of place cells in the hippocampus, they could reconstruct the location of the rat to about 3 centimeters. So if you, for example, say that there is some activity in this cell, some activity in this here, some in this, then probably the rat is somewhere here at the upper corner, and there would be very little or no activity in those place cells, but in these here, but there would be activity here and here, of course. So out of knowing the where these place fields are, where the place cells fire, you can reconstruct the location. Now, one interesting thing about the firing of the place cells is that it interacts with the theta oscillation in the hippocampus. So theta is an oscillation at about 7 Hertz, so 4 to 8, 4 to 10, I think, but around about 7, which goes like this. So, and it gets plotted in 2 different ways. So it tracks the cumulative activity of all the pyramidal cells in the hippocampus, but you can also plot it as a strength of inhibition. So here it is plotted as the cumulative, the total firing of all the pyramidal cells, so the excitatory cells in the hippocampus, the place cells. And where this is down, that is where the place cells are not firing, where inhibition is strong. Sometimes it gets plotted the other way around, that up on that curves means the inhibition is strong. So these are complementary. If one is strong, the other is weak. So here in this case, you plot the total activity of place cells, of all place cells, and you do that as a rat walks through at a steady speed through this place field. So this here shows the firing rate of that place cell as the rat walks through. So here that is shown in color. So you imagine, for example, that you walk through here. There's no activity, a little, then it gets more intense. So the red here is a higher firing rate, the yellow lower again, the blue lower again. So this here would be blue, yellow, red, yellow, and blue again. And if the rat walks here at a steady speed, then it will experience a particular state of the theta oscillations at each of these locations. And here, where this place cell has a low firing rate, so this is at the edge of its place field, so when the frequency is low, the place cell hasn't got a lot of input, and therefore it can only fire, because it has weak activity, weak input, it can only fire when inhibition is really, really weak. So it can only fire at the point in the theta cycle where inhibition is minimal. So if you were to plot that, this phase and position, so if this here corresponds to this here, that is just basically a place cell that is further along the line, then the firing can only happen in this narrow time slot when inhibition is minimal. Then inhibition increases again, and while the rat is walking through this part of the place field, the cell can't fire. So that would be this range here, So during this part of the theta cycle, that is this part here, the cell can't fire. Then the rat has now reached this part of the place field. By the time the inhibition decreases enough, oh, sorry, yes, that is plotting inhibition, not the excitation. So here, the inhibition has decreased enough that the cell can fire again, but because it's further into the place field, it has a stronger input, it has more activity, it can fire slightly earlier. So it's a bit before the minimum inhibition. So it can fire a little bit earlier, which here corresponds to something like this. So the earliest firing is here, but also it can fire through a longer span of time, through a longer interval. That is why there is more scatter here over the firing relative to the phase of the theta cycle. And the same again. So then inhibition increases again. At this point, the cell doesn't fire anymore. So this firing here, this theoretical firing rate, you don't see. Inhibition peaks. Then this part of the theta cycle, inhibition decreases again. And once more, you have then stronger activity on the part of the place cell. Then the first firing comes earlier in the theta cycle, so it's shifted here, and that means that you get earlier firing in the theta cycle, and again a longer time interval through which the cell is able to fire. So it's now a time interval from here to here, so a larger part of the phase, and so on. So here you get to the point then where the firing can happen very early, and where there is a very wide range of moments in which the cell can fire. And this here is then another plot of the same thing, and it also then shows if you correlate the phase with firing rate, because here the cell can only fire through a very narrow time interval, then you have a pretty good correlation with the of the first firing with phase. Now, how do you get these oscillations in the first place? So how do you generate them? And here is a model that shows this. So in this case, the idea is you have an oscillating input which already elsewhere the theta oscillation has been generated. And here it shows how you can get another oscillation on top of that, the gamma oscillation. So imagine you have a steady input, say on this neuron and this neuron. So these two get input, these two here get nothing. So they get input here, they fire. Then there are inhibitory neurons, which are widely connected. So if you think back to here, there were a few cells here which had firing fields that were not location specific. And these, when you then check, are usually inhibitory interneurons like this one here. So they're not place specific. They connect to lots of place cells, therefore to lots of cells that fire in different places, and they will fire in response to any of those. Therefore, the firing of those cells can't be place specific. So they collect input from everywhere. Then they feed back to here, and that is an inhibitory connection. And so what that does is, if you have a steady input here, you have a bit of a delay, because after all, signals have to travel along here. They have to reach this cell body. That cell body has to depolarize. It has to send out an action potential. You need time for the synapse to function. And therefore, you've got delayed inhibitory feedback. That feedback then shuts off the input. So if only these two neurons are getting input, that is shut down then. then there is no input left for this inhibitory interneuron. So at that point, the inhibitory input shuts down this here. That then shuts down the pyramidal cells. That then deprives the inhibitory cell of input, so that closes as well. That means the inhibition goes away, and that means that this input can go through again. The whole cycle starts through again. And so if you have delayed inhibition, then a steady that feeds back to where the input comes in and shuts down the input, then you can turn a steady stream into an oscillating stream. And the frequency of the oscillation will depend on how strong these synapses are, how large this cell body is. So if it's a larger cell, then it acts like a large capacitor and it takes longer to depolarize. If it's a small cell, then it's faster to depolarize. And again, on the strength of these synapses here. And it so happens that these inhibitory cells are working a bit faster, and so you get a relatively fast oscillation in the range of 40 to 120 Hertz, whereas the theta is in the range of 7 Hertz. And the idea then is that out of this circuit, you can actually make a short-term memory. It's not a working memory because there's no processing going on, so this is only a model of short-term memory. Then this kind of anatomy can give you a long-term memory network, where if you have, for example, information coming in here and here, but nothing here, that feeds back. You then have simultaneous activity from the input and from that recurrent connection. So from this one that goes to here to both of those, and from this one to that goes to here and again to both of those, well, actually to everything except possibly itself. Then if you apply the Hebbian learning rule, things that fire together, wire together, that reinforces these connections. And then if you give even just part of the input, say this one here only, so this pyramidal cell fires, you have this here coming through, that then activates this one here. And so with even a partial input, you can get the whole input back. And so the point of a recurrent network like this is it gives you a way of implementing pattern completion. A colleague of mine once set the exam question about memory. So it's not a multiple choice question where you can just say, this is a complete thing, and all I need to do is find out, have I seen this before? You get a minimal retrieval queue, and you have to complete the pattern. And so an auto-associative network like this with recurrent connections, recurrent collaterals, is one way in which you can build a long-term memory system that does exactly this job of pattern completion. And it turns out that you can put these two networks together, that if you superimpose them on each other, you can have a network that simultaneously implements short-term and long-term memory. So if you keep things cycling through this part of the network, this short-term network, then you have input that can transfer information into long-term memory. And the way that works is, so imagine that you have 10 cells. So not just the 4 that are shown here, but 10. And you have an input coming to some of them. And so this might be something you see, this might be something you hear. It gets translated into a pattern of activation. So this here shows that pattern of activation. This here shows the membrane potential of the corresponding pyramidal cell. So that gets depolarized, and the idea is that this depolarization triggers off what's called an after depolarization. So that's a current flowing into the cell, and hyperpolarization here, that depolarizes the cell until it hits the threshold and triggers another action potential. So if we go down here, also, if this input happens to arrive so here now theta is plotted the other way around. That's part of the reason why I got confused, because I remembered that theta is plotted in opposite ways in different pictures I've got here. So here Up means high activity for the pyramidal cells and low activity for the inhibitory cells. So basically here, when this here is up, that means inhibition is low, so the opposite of what we had before. While here, the pyramidal cell activity is low. So because we have here strong inhibition, little activity of the pyramidal cells, that's why the cell isn't firing straight away. What we may get, though, well, if information goes through, we can get something in the inhibitory cells, but that really should only, I'm not actually sure why it's here, because it really should be happening only when there is an action potential. So what we then get is when we are at this moment in time, we always need to think about things happening, these several events happening simultaneously. So then we progress through time, and what we then get is this current flowing into the cell and slowly depolarizing it. And we have also gradually more activity of the pyramidal cells as inhibition becomes weaker. So then again, more depolarization and more. And this here is the point then where this cell has depolarized enough that it can fire at this point in the theta cycle. So it fires, that gives an action potential, and that then triggers here a current in an inhibitory interneuron. So now here we've got a response from an inhibitory interneuron, which again shuts off everything. So these things here should have happened to all these 5 cells. So even if we don't sustain the input, even if it's a one-off input, this after depolarization in each of those 5 cells should then make sure that those 5 cells fire again at about the same time, thanks to this after depolarization. And if there is no other input, then they will fire basically as early as they can. So this here would be the earliest part of the theta cycle where that firing is possible. Right? And then the same thing happens again. As you go through here, you then have again this after depolarization. It now starts relatively early, and because it now has that head start, it can actually build up to a higher level, and therefore this whole thing now shifts to a little bit earlier in the theta cycle. So previously we had it only at this point in the theta cycle, already fairly close to the peak. Now here it's less than halfway from the trough to the peak. So now we get our action potential here. We again get then activity in the inhibitory interneurons, which shuts down everything else. And again, we get after depolarization. So that then carries on. Here then we have a second input. So now it's a different bunch of pyramidal cells that get input. And it starts off their depolarization. And they happen to get their input again when inhibition is strong and when the pyramidal cells can't fire anyway. So then we have this first set of cells firing again because they started their after depolarization earlier, and therefore they fire fairly early in the theta cycle. Then we have here the inhibition. And so during that inhibition, no other pyramidal cells can fire. Then this after depolarization has increased to the point where at this point in the theta cycle, where inhibition is really quite weak, again cells can fire. And so now you have basically this here first, this here second. So you have now 2 time slots occupied, and so on. And then it keeps going this way, and the 2 different patterns, the 2 different items, even though they're in the same network, they kept separated in time, and that works because of that gamma oscillations. So each of those patterns is firing during its own gamma oscillation, and the next one comes in the next peak of the next part of the gamma oscillation. And then if you look at putting other pieces of information in. So here you have the first slot occupied in the theta cycle. Here you have the second slot occupied, the third slot occupied in the theta cycle. You put in another piece of information. It then gets moved to the first available slot. So that is how this network works. And then basically what you end up with is the capacity of the system is as many patterns as you can fit into the active part of the theta cycle. So according to this model, the working memory capacity or short-term memory capacity is limited by how many gamma cycles you can put on top of a theta cycle. So that then gives you this idea of a short-term memory network and a long-term memory network superimposed into a single network, where short-term information just keeps cycling through those cells thanks to the after depolarization and its interaction with the gamma cycle. That then sends out information that sends it through here, through these recurrent collaterals, where information can be transferred into long-term memory. Now, one important thing is here about long-term memory. So learning is then by modifying the strength of these synapses. And that, of course, is a slow process. And so if you need something faster, you really need to do something else, which I'll get to in a moment. So here, coming back to that point about the capacity, you then have pattern 1, pattern 2, and so on, in this case up to pattern 7. So the idea then is this here, from here to here, that's the time interval that corresponds to the theta cycle, and the gamma cycle, and this is how many gamma cycles you can put on top of the theta cycle. And also there are two different kinds of NMDA receptors, fast and slow. If this network here uses fast NMDA receptors, then this activity here dies down before the next pattern comes in. So this is a way of putting one pattern into that long-term memory network and keeping it separate from the next pattern. While if you have a slow NMDA receptor, then the channels are still open when the next pattern comes in. So basically you would have here some synapses still, some of the postsynaptic channels still being open to input, and therefore when a new pattern comes in and is cycled through the system, this synapse can still be strengthened. And so what then happens is you learn sequences of things. You associate successive patterns with each other. But as I mentioned, this modification here, this is relatively slow. So if you need to control the flow of information by modifying synapses, that takes time. And there are occasions when you really need to regulate the flow of information a little bit faster, because after all, you can focus on one thing, you can exclude other information, you can switch between streams of information. Then the question is, how do you do that if you don't do it by changing the strength of synapses? Now, you might be able to do it by inhibition and by fairly targeted inhibition, but of course the problem there is that then if you don't want some information coming from some part of the brain, if that's not useful in a particular part of the brain that can receive it but hasn't got use for it right now, then if it sends inhibition back to that point of origin, that of course would shut down everything if it goes all the way back. So then it's and that part of the brain couldn't send information to yet other parts, and that might not be a good idea. It might still, that information might still be needed elsewhere, just not right here. If you want to inhibit in this fashion here, that you shut down specific inputs, then of course you would need to know exactly which inputs to shut down, which might also be difficult. So there is another way of doing that. And this also matters to, for example, schizophrenia, because one idea is that schizophrenia, or more generally speaking, delusions and hallucinations, are connected to this flow of information. And that is by the idea that we recognize the consequences of our actions through prediction. So if you look here at how do we know whether an object is moving or not. So here you have the situation, the eye isn't moving, the object isn't moving, everything is stationary on the retina, you conclude that the object is still. If the object is moving, that creates motion on the retina, but you have to distinguish that from this case where you get exactly the same motion across the retina, but you get it because the eye is moving. Now, the idea then is you know that the object is still and the eye is moving because you've given the command to move the eye. And experimentally, you can check that by either passively moving the eye, so there hasn't been a command, and then if that idea is correct, that if you basically subtract the intended movement from the experienced movement, here you didn't intend anything, so you don't do the subtraction. Therefore, in this case, it should look to you like the object is moving. And also, if you interfere with the movement, for example, by injecting the eye muscles with curare, and you intend a movement, so then you give the motor command, and you do the subtraction. So basically you say, I'm going to experience this much movement, I have to subtract that from what I actually see. Well, you subtract it from 0, therefore you get some experience of movement. And the idea then is that if you get a large enough difference between what you predict and what you experience, then you assume that there is some other cause to your perceptions, and this is how you distinguish between what you're doing and what happens otherwise. That is this notion here of a motor command. You make a copy, you make a prediction, you compare that to what actually happens, and if there is a large enough external influence, then you will get a difference between that, and that is how you actually conclude that there was an external influence. And if you do that, of course, you need to send information between brain areas that create commands and brain areas that then monitor the sensory consequences. That's the idea of the forward model. And there is a finding that when you compare your normal controls to non-hallucinating schizophrenia patients and to hallucinating schizophrenia patients, and you look at the coherence between when you're listening and when you're talking, for the controls, there is a clear difference in the coherence when listening and talking. For the non-hallucinating schizophrenia patients, there's a lot of variation, so things are unclear. But here in the hallucinating patients, there is no difference. So, and the question is, what exactly is measured by that coherence? And the idea then is, so imagine here is a brain area that is sending information to here, but it shouldn't send information to here. And one way in which you can achieve that is by synchronizing these oscillations. So you create, for example, an oscillations through this delayed feedback mechanism, and you do that in each of those brain areas, and you have a way of either synchronizing them or desynchronizing them. And so if you're here in the part of the cycle which permits activity, then of course you can combine information from here to here or from here to here, so they can influence each other. While here, if the information arrives with, of course, a little bit of a delay, at the moment when here the inhibition is strong, then it simply can't do anything, so it will get lost. And that way you can have a regulation of information flow. by either synchronizing or desynchronizing this activity, or even just not having coordinated activity in the first place. So then you would have some information coming through, but it would be pretty random. It would be less effective. And so what's being measured by the coherence then is coordinated activity. Is activity coordinated between these two parts of the brain? And then you can tell how the information flow functions by looking at both coherence and phase. So if, for example, you have information going from some subcortical area to two different cortical areas, what you should find is that the subcortical is slightly ahead in time, because these action potentials, it takes time for them to travel along the axons. And if instead you have that relationship here, that this here is earlier than this here, and you know that they are that there is coordinated activity, then it's probably input coming from here to here. So in both cases, there is coherence between these two cortical areas, but if they're absolutely simultaneous and there is enough distance between them that there should be a delay, then the coordination comes from a common cause, namely input from elsewhere that they're both receiving. While if one of them causes activity in the other, then you should have a phase shift and coherence both. There are different ways for brain areas to interact, and this here is from the paper by Jensen and Colgan. So they say, let's say you've got here some theta, and the theta may actually change a little bit in amplitude, so it's a little bit amplitude modulated. And they propose that there are theoretically 4 different kinds of coupling between a theta oscillation and a faster gamma oscillation, and they provide references for 3 of them. So 3 of them have so far been found. But one thing I should say straight away, I have no idea what the functional differences are between these different kinds of coupling. So I have not followed that up. I haven't got a clue. I just read in Jensen and Colgan that these exist. So one is power to power, where if you look here at what's the amplitude of the theta oscillation, so that varies a bit, that the amplitude here of this gamma oscillation, even though it has a higher frequency, but the amplitude varies in the same way. So you have here amplitude modulation in both cases. And so that is called power to power. When the amplitude is large here, it's also large here. When the amplitude is small here, then it's small in the theta as well. Then there is phase-to-phase coupling, where you have a fixed phase relationship, so that you have, for example, from here to here, there is so many milliseconds delay, and again from here to here, so many milliseconds, and again from that peak to that peak, from that trough to here, and so on. And the way that then works is you really then, if you want that fixed, you really need one of the oscillations, the faster oscillations, to be at a multiple of the frequency of this one here. Then another kind of coupling, and that is the one for which Jensen and Colgan don't provide a reference. So at the time they wrote the paper, I suppose this hadn't been found, or at least they don't know about it. That is phase to frequency, where when the amplitude here is large, then you have a higher frequency. When here the phase to frequency. So when you hear up, then it's a high frequency. When here you're down, a low frequency. Here when it's up, high frequency. Here when it's down, low frequency. And this here is phase to power. And so that you can get when you have the slower oscillation modulating inhibition. So here the theta cycle permits activity, so you have strong activity in the gamma cycle. Then here, when the theta cycle inhibits everything, then you have also weak activity here in the gamma oscillation. And again, here activity is permitted, so then you have again large amplitudes. And so this is what we saw in that theta gamma interaction in that model of short-term memory, where the gamma was shut down during parts of the theta cycle. And so that is basically all I've got. So the EEG oscillations come from many neurons working in coordinated ways, firing in a coordinated manner. You can get that coordination, that synchronization, from inhibition with delayed feedback. Depending on the properties of these cells and how long the delay is, you then get oscillations at different frequencies. You then can use those oscillations either to coordinate information flow, to determine where information goes, by either choosing to synchronize or not, by getting the phase into a particular relationship or into a different relationship. And you can do interesting things by having different oscillations interact, like this model of short-term memory.